So in this last recording, the last recording of this section on cells, let's look at how cells reproduce. And that's going to involve a process called mitosis. So some of this is going to be nuts and bolts. We just got to get these processes down. And if we do it well now, it's going to pay off big time as we start to talk about genetics more later. But we know based on the cell theory that cells only come from other cells. And this is really interesting because it means that there's an unbroken line of life, cells coming from other cells, for about 4 billion years. It's a really long time. It's really interesting. And for a good portion of that time, cells were only single cell. There weren't any multicellular organisms. So the act of dividing and copying a cell was the means of reproduction. And if you're a single cell, that's it. That's how you reproduce yourself. Now, if you are a multicellular organism, each one of your cells can divide to help maintain and grow and develop the body, right? So, for example, think of your skin. So actually, the very outer layers of your skin are dead cells that are constantly being shedded and sloughed off. And so those have to be regenerated. So we have cells down in the dermis and in the lower layers of the epidermis that are dividing. Right? So those cells have to reproduce to maintain your skin. So more even than just that maintenance, it's growth and development. Right? So we all started, and we'll learn this later, we all started as a single cell, and now we're somewhere between 1 and 10 or 100 trillion cells. Right? The bigger you are, the more cells you have. And in addition, we can heal ourselves to a certain extent. So if you get cut, those cells get turned on to start to divide to heal the damage of the body, right, if you're a multicellular organism. So cell reproduction is a big deal. If you're a single-celled organism, that reproduction, that is your method of reproduction. If you're multicellular, oftentimes you have a means for sexual reproduction, which we're going to talk about later. But you have to grow and develop. That's going to be rely on cell division. Okay, cell reproduction. You're going to heal, cell replacement, and then ultimately we are going to study how in sexual reproduction you produce special cells through a special kind of cell division called meiosis for the exchange of genetic materials. But for now, let's just think about what's called asexual reproduction, where we're creating clones or exact copies of the cell that we start with. So we're going to start with the simplest kind of cellular reproduction, and that is the reproduction in the simplest kind of cells, and that is prokaryotic cell division. So again, we are going to come back to the themes that we've seen earlier in the quarter, constantly building on what we've learned before. So prokaryotic cells are simple cells, one-room studio apartment. That is going to lend themselves to their greatest strength right here, and that is their ability to reproduce very, very quickly, right? So the speed at which a prokaryotic cell can reproduce itself under optimal conditions can be on the order of 15 or 20 minutes. Now, to compare and contrast that to kind of the optimal conditions for a human eukaryotic cell, we're looking at 12 to 24 hours, right? So this is the advantage of being small and simple. And the process then, which we call binary fission, right? Cell division in prokaryotes, called binary fission, 2 splitting apart, is also going to be pretty simple. So one of the things that's important in reproducing a cell is making a copy of the blueprint. And so the DNA is the blueprint. So the single circular chromosome that we find in prokaryotic cells is going to make a copy of itself. And those 2 copies are going to separate from each other, moving towards the opposite ends of the cell. As the cell elongates, then ultimately we'll start to pinch off in between those 2 DNA copies until we have 2 new cells. Boom, we're done. That's it. So again, under optimal conditions, this can happen very quickly. 1 cell can become 2, becomes 4, becomes 8, 16, 32, so on and so forth. And this exponential growth can lead to very, very overwhelming numbers. And that is ultimately how prokaryotes survive. We can talk about that idea in details more later. So prokaryotic cell division is called binary fission. Eukaryotic cell division, of course, is going to be more complicated. Surprise, surprise, bigger cell, more complicated cell division. All right? So eukaryotic cells have a lot going on inside them, right? So here's our picture of our animal cell. Lots of organelles, lots of things going on, many compartments. The plant cell, we have our cell wall, central vacuole, chloroplast, as well as our mitochondria. There's a lot going on, right? But the key to cell division is going to follow the same template. We need to make a copy of the DNA, of the blueprint, to make sure each daughter cell has that information to run the cell. So what we're going to focus on is the process of the cell getting ready, and then spend more time on the details of making sure that the nuclear contents are evenly and systematically distributed to the new daughter cells, and that's going to be called mitosis. So what we have to do is go back and review kind of the DNA in the nucleus. So remember, we looked at DNA briefly, that double helix, and in eukaryotic cells we talked about it being packaged with proteins. So what we're going to be able to do is take that lots and lots of DNA and wrap it up so it can fit inside the nucleus. But we can condense that even further into what we call metaphase chromosomes, or condensed chromosomes. So a chromosome, again, is just a big piece of DNA, whereas prokaryotes have but one chromosome that is circular. Eukaryotes have multiple chromosomes and are linear. And at the same time, they're going to make a duplicate of every chromosome. They're connected here in that metaphase chromosome. That's why it looks like an X, and we'll talk about that more later. But we're going to condense that down into a package. It's like taking all the stuff in your kitchen and putting it in boxes, right? I'm going to put it in boxes so that I can move it very carefully to make sure I don't lose any pieces. And so that's what we're going to do with our DNA. We're going to package it all down into this condensed chromosome. And again, laying down the foundation of understanding mitosis and eukaryotic cell division is going to allow us to understand genetics much better later. So I'm going to go over this with you now and look at this condensed chromosome. So the very top of this slide, you see that purple blob, elongated blob. That's one chromosome. Now, because we're going to make two daughter cells, we need two copies of that chromosome. So we're going to look at that second level here, where we see two sister chromatids, which are connected by what's called a centromere. That is also one chromosome. So that's a duplicated chromosome. That duplicated chromosome will split into two during cell division, and the two daughter chromosomes then will go in opposite directions to each of the daughter cells. So you have one chromosome at the top, one duplicated chromosome that will split then into one chromosome and one chromosome going in opposite directions. Okay? Hold on to that for now. We'll go over it, and it will come into play later. So how does a cell prepare to divide? Well, what we have is called interphase of the cell cycle. So the cell cycle is a term that we're going to use that refers to a cell that is actively dividing, right? So these are cells like in your skin that have to be constantly dividing to replace cells. Not all cells are in the cell cycle. Some have kind of gone into what's called gene knot, which is kind of a holding pattern. They're kind of just doing their job. They can reinitiate oftentimes the cell cycle if needed. When you're growing, certainly the vast majority of your cells, if not all, are in the cell cycle. So 90% of the cell cycle is this big blue arrow that wraps almost all the way around this clock-like picture, and that is called interphase. Now, interphase is going to be all about preparing the cell to divide. So the first part of interphase is called G1. And G1, the G stands for gap. Because if you look at the cell under a microscope, it looks like nothing's happening. It looks like there's a gap in activity, when in actuality there's lots and lots of things happening. So G1, the cell is growing. It has just come out of cell division. It's fresh. It's a new, fresh daughter cell. It's come out of a division, and it's growing. It's making more organelles. It's making more proteins. It's getting its biochemistry up and running. It needs to get all the machinery ready because it's going to go into S phase next, which S stands for synthesis, where it's going to make a copy of all this DNA. We need to make a copy of all the DNA because we're going to send one copy to one daughter cell and the other copy to the other daughter cell. We've got to have the blueprint, the instructions for the cell to run. So once we've gone through G1 and S, we go into G2, gap 2. Gap 2 is now the cell preparing, getting all the proteins made and the machinery to go into M phase, or mitosis. Mitosis is going to be about partitioning the nuclear contents, i.e. the chromosomes, into each of the daughter cells. Very important. Now remember, all of this is being controlled by enzymes, proteins that are catalyzing reactions and controlling the biochemistry of the cell. Now, as we finish up G2 and enter mitosis, we can actually see the cell changing under a light microscope, and we're going to be able to follow those stages by looking at pictures of the cell. Again, mitosis is a stage where you kind of have everything packaged up in your house. Now you're going to put it on the truck. Nothing is accessible until we get to the final destination. In that case, will be the new daughter cell, and then we'll unpackage everything again, blow it up. Okay? So let's take a look at the M phase, or mitosis, stage by stage. So these are pictures that you're going to need to be familiar with. You'll need to be able to identify each stage of the cell cycle. And so that only comes with practice. So let's start by introducing them, and then we'll spend time practicing that later. So let's look at our first picture here. We can see G2 of interphase. So we see the DNA in the nucleus. It's in its chromatin format. It's like spaghetti in a bowl. The DNA is packaged with proteins, but loose enough that we can access it. It can be read, so it can be used to direct the cell. And it's getting ready to go into M phase. Now, M phase, or mitosis, is going to be broken down into 4 stages, and I call them PMAT. So prophase, metaphase, anaphase, and telophase. And this is a continuum. We're going to see individual pictures, but really it's a flowing process that you're going to have to make a best judgment call when you see a picture of a cell. I'm going to give you criteria for each stage. As we enter mitosis, the act of distribution of the reproduced nuclear contents, prophase is going to be the first stage. And prophase and prometaphase, as you see in this slide, you'll read prophase, metaphase, and other in the book perhaps, but I'm going to lump this all into prophase just to keep my PMAT nice and clean. So PMAT prophase is the first start. PMAT. P for prophase. In prophase, we're going to have a couple things happen. One, the chromosome is going to condense. Now, because it's been duplicated in the S phase of interphase, it's going to condense into that kind of X format, where you have 2 what are called sister chromatids. Each chromatid is a copy of that chromosome. Now, we, as humans, have 46 chromosomes. So whereas this picture shows only 4, if this was a human cell, there would be 46 of those being condensed and then starting to line up. Right? So the chromatin will condense into condensed chromosomes. Now, the second thing that happens is the nuclear envelope, the membrane that makes up the nucleus, will start to break down. And you can see that here as we go from prophase to prometaphase. So this happens in prophase. The nuclear envelope starts to break down because we're going to have to make room and get these 2 copies of the DNA separated from each other. We have to get them out of the room that they're in. That's the nucleus. 3, the mitotic spindle starts to form. So the mitotic spindle is microtubules. They're components of the cytoskeleton. They're long protein tubes. And they will form this network that will spread out across the cell, and they will attach to the chromosomes, physically moving the chromosomes around. And that is the 3 big things that happen in prophase. Chromatin condenses into our condensed chromosomes, the nuclear envelope breaks down, and the mitotic spindle forms and connects to the chromosomes. Our next phase in PMAT, P-M-A-T, is metaphase. And in metaphase, those chromosomes attached to the mitotic spindle will then be positioned on what's called the metaphase plate. And this is an imaginary slice through the middle of the cell on which all the chromosomes will line up. This is very important because if all the chromosomes are lined up on the metaphase plate, when we go into the next phase, anaphase, we're going to pull apart those chromosomes and move in opposite directions from the metaphase plate. So again, in metaphase, PMAT, prophase, metaphase, anaphase, telophase. In metaphase, lined up on the metaphase plate. Anaphase, the mitotic spindle will then pull the duplicated chromosomes apart. So one chromosome then becomes 2. One of those each chromosomes that are now formed from the split chromosome move in opposite directions, moving towards opposite ends of the cell. So that in telophase, we can reverse the process of prophase. So in telophase, now I have one copy of all the chromosomes at one end of the cell and another copy of all the chromosomes at the other end of the cell. Then we can decondense those chromosomes, reform the nuclear envelope, and start to break down the mitotic spindle. We don't need it anymore. That is the exact opposite then of prophase. So telophase is the opposite of prophase. Now, in addition to that, the actual division of the cell can now occur because I've separated two sets of instructions apart from each other, and we can split the cell in between those instructions. So we call this cytokinesis. So in animal cells, we have what's called a cleavage furrow forming. So you can literally see there are microfilaments, another form of the cytoskeleton, forming a protein ring underneath the plasma membrane. It will literally start to cinch down on the plasma membrane, snipping it in two, and we get two new daughter cells, each with a brand new nucleus, exact copies of the parent cell. In a plant cell, we have what's called the cell plate forming. So the cell plate then is vesicles full of cell material fusing in between the two different nuclei, forming a new cell wall in between them. Two new identical daughter cells. Right? So again, if we look at the process of mitosis, the distribution of those nuclear contents, and then cytokinesis, we can see actual pictures from microscopes here. So interphase, you see a normal cell. You see it entering prophase, condensation of the chromosomes, breakdown of the nuclear envelope, formation of the mitotic spindle. Metaphase is the easiest one to nail because they're all lined up on the metaphase plate. Anaphase, they start to pull apart. That's probably not the best picture of anaphase, but you know, early telophase, I'd definitely call that. Late anaphase, early telophase. And then late telophase, you actually see cytokinesis has already occurred, and the chromosomes are now starting to decondense, and new nuclear envelopes are starting to form, and the mitotic spindle has been broken down. So you should be able to look at each of these cells, prophase, late prophase, metaphase, anaphase, telophase, post-cytokinesis, and be able to identify those and be comfortable with those steps of the cell cycle.